So we have the sequence data here, and now I'm going to talk about how do you turn something like this into numbers, because I like numbers. So there is a general pipeline to do that, and we're assuming the first 3 steps are like what a biologist would do, and obviously there's a lot of intermediate like quality control steps and like details, but I just want to get across the general idea and framework of how it turns into numbers. So the first 3 steps would be like the biologist taking the mRNA and then preparing it and getting it sequenced, and then raw reads is where we're at right now, where we have millions and millions of lots of fragments that are sequenced. So the next step we want to do is alignment. So we have these fragments, and if you remember, mRNA is a copy. We went from DNA to the mRNA. So alignment is we have this like now sequenced, we want to find out where that corresponds to the DNA again. Like where did it come from? Which can be tricky because of like processing steps between, but hopefully software will help us with that. So if you have a reference genome, like if you're working with an organism where the genome is already sequenced, like humans, we had the Human Genome Project, we have the sequence already there. There's also a lot of species too that have the genome sequence, like zebrafish, Arabidopsis, fruit flies are common ones. So what you would do is you take the RNA fragments and then try and find where it lines up. The top rainbow thing up there, that would be the reference genome. And you try and find out where your fragments match. And what you're looking to do is the genes that are expressed a lot will have a lot of reads corresponding to them. So like something over here where there's lots of things stacked up, that gene's being expressed a lot. But if you had something like here where there's, I think I only count like 4, that means this part of the genome is not being expressed as much. And you get like some sort of, you see the blue like little distribution curve of gene expression. So that's a lot closer to numbers that we're looking at. If you're working with an organism whose genome isn't sequenced, you can still try and do genome assembly and get through this pipeline. It would still work, but you'd have to go through a different process. Like obviously you couldn't slide it across the reference if you don't have a reference. I'm not going to get into the details of that, but it is a thing. So obviously, like, I don't want to do this by hand, so I want software to do it for me. And these are all 3 software packages you can use. They are free. The most popular one is Top Hat, or Top Hat 2 now. And one of the reasons it's most popular is it's part of this Tuxedo suite where they have tools to do this entire process all in one. So like the output of one automatically feeds into the input of the next step, which is kind of nice. And it has a nice name, Tuxedo, because there's bow tie, top hat, and then the next one's cufflinks, and then you can use cummerbund after that. So I'd like to think that has to do with its popularity. Maybe, I don't know. Another thing is GSNAP, which is better at detecting novel transcripts, and it also deals with SNPs pretty well. I've heard that it's slower in some instances. STAR is another one, and that is like, it's designed to be the fastest one, and it does take up more RAM though, but if you had like a really, really big sample and it's important to you that it's fast, STAR might be a good choice. So let's suppose we ran our software of choice and we aligned it to the reference genome. The next step is to do the feature counting. So we want to go through each gene and then count how many overlaps we have, and try and get this distribution of gene expression going. So there's 2 ways you could try and go about this. Like the first one, you could just straight up well, I wouldn't do it, but software would like just get the raw counts of how many overlaps we have for a gene. And two common packages to do that type of strategy is one's ht-seq count, that's based in Python. There's also feature counts, and that's an R package, and it is the faster one and uses less memory. It's faster not because R, but because it's a wrapper for a C function, and it's been designed like to just, let's make this as fast as I can. The other strategy you could do is, let's say you don't want to just go out and count all of the overlaps. Maybe instead you want to estimate the counts based on like the probabilities that you estimate. So one algorithm that does that is RSEM, which is actually an expectation maximization algorithm. So they've created like some sort of likelihood where each parameter is like the probability that these fragments will overlap at a specific place, and then they use the algorithm to like estimate it. If you want to read about the details of that, like I did put the paper at the end with all the exact details. I just thought it was cool because EM, I know that. Cufflinks, I mentioned that earlier as part of the Tuxedo suite. So after using Top Hat, you would use cufflinks to quantify the transcripts. There's also a lot of other ones that do this kind of estimation of the counts. The estimation could be good if you don't have a reference, like, because you could get around actually having to align it. And also, if you're not doing the alignment, it could be faster for you. So if for some reason that's something important, this might be the better way to go.